Hey guys, and welcome back to SciSci. In this lesson, we're going to be talking about gel electrophoresis and continuing our series on research techniques. Now, our last video was on tissue culturing, specifically on how to split your cells and maintain a tissue cell line. This topic is going to have two videos. One, in this video is going to be on the function and how gel electrophoresis works. And the next video is going to be me in the lab showing you exactly how to do this procedure. Well, let's get started. In addition, protein gel electrophoresis requires a video of its own, so we're going to be talking about that in a later video. Now, let's get started. DNA is negatively charged, and this is going to be a fact that we need to remember throughout this video. DNA is negatively charged. Now, if you want to go back to our last video, to a past video that was talking about nucleic acids, you can check that out and understand exactly how DNA is negatively charged. But in essence, it's because there's negatively charged phosphate ions on the backbone of our DNA. But this fact is going to be the fundamental reason how our DNA gel electrophoresis. Before we get into how it works, let's talk about the function and why this is important. Well, DNA gel electrophoresis helps us separate DNA based on size. It helps us analyze genes and compare DNA samples. Now, there's multiple real-world uses, but some examples are, in the case of analyzing genes, you can identify if someone has a hereditary disorder, and in the case of comparing DNA samples, you can see if someone has the same DNA sample as one found in the crime scene, or see if two people are related. Now let's get started on how this actually works. Now what I've drawn here, at the top you have a block, and that is our power source, and that's going to help us create an electrical gradient. And at the bottom here we have our gel electrophoresis apparatus. From the right side to the left side is going to be TAE buffer. So from the right side to the left side is going to be TAE buffer. And you're also going to have a central area, and that is where you can see there's a blue box in the middle of our apparatus, and this is our gel. Now this gel is also going to be submerged in our TAE buffer, and this is going to allow us to have an electrical gradient from negative to positive across our gel electrophoresis apparatus. Now we have a power source and TAE buffer that allows us to have this electrical gradient. But what is this used for? Well, as you can see in this drawing, you have our DNA in this well, and it sits really nicely within this well in the gel. Well, continuing on, so you have this well in the gel and you have a DNA sample in each of these. So you're going to be using a micropipette, help us insert these DNA samples into each of these wells. Along with this DNA sample, we're going to be having glycerol as well as a dye. Now, the glycerol helps our DNA sit at the bottom of the well, and the dye helps us track the DNA as it moves across. But why does our DNA move across? Well, it's pretty simple actually. You have our electrical gradient, and we know that DNA is negatively charged. So it's going to be attracted to the positively charged side of this electrical gradient, or the farther side of the gel. And so when we turn on our power source and when we have all of our DNA samples within this gel, we're going to see our DNA samples move across the gel. But how do we separate the sizes? And this is the next step. As you can see here, I've kind of created three different cross sections throughout this gel. And we have our large, medium, and small sized DNA samples. And in this case, I've kind of wrote out exactly the size of the DNA sample. So you have an 8,000 base pair sample, a 2,000 base pair sample, and a 750 base pair sample. Now, it's not necessarily that these pores are larger or smaller throughout this gel. Instead, it's that the longer DNA strands take a longer time to move across the gel and move through all of these pores, while these smaller DNA samples take less time to move across. And so, once you have your smallest DNA sample reaching about 90% of the way, down your gel, the rest of the DNA samples of different sizes will line up behind it based on how large it is. And so the medium sample would be in the middle at around the 2000 base pair area, and your largest sample would be near the beginning because it takes much longer to go through the gel, and it'll be in an area that'll be about 8,000 base pairs. So when we look at our gel, and when you look at your own gel, it's important to remember that it's not necessarily exactly where your DNA is on the gel that decides what the size is, but instead you're going to be having a DNA ladder that I'm going to be showing you when we in our next video. That DNA ladder will have different base pair sizes, and you can see and you can compare based on that how large your DNA samples are. Now, going back, I mentioned that our gel has about the same pore size throughout. And in this case, our gel is made up of TAE and agarose. And TAE is the same thing as the buffer, so it helps maintain that electrical gradient. And then agarose helps solidify the gel and helps us create those. Now, a general gel size is about 1%, so you have 1,000 milliliters of 1 times TAE, and you also add a gram of agarose. When you mix these together and microwave them or heat them and then let them cool, you can actually get a solid gel. And so that's how we create our gels. And that's kind of the essence behind how we also maintain that electrical gradient throughout the gel itself. So we've talked about how you can separate. So we've talked about why our DNA passes through the gel, and that's because it's negatively charged and you have our electrical gradient. We've talked about how those sizes are split up because larger strands take a longer time to go through and smaller strands take a bit less time, and so it can get through the gel much faster. But let's talk quickly about a use for this. So let's say you have our ladder. Now in this case I haven't drawn the ladder. The ladder would be in its own well, but I've labeled it here to kind of help us understand. And this is our sample number 1. This is our sample number 2. This is our sample number 3. And for the fun of it, let's make this example a crime scene. So you have our DNA sample from the crime. If you see here, you have similar DNA samples from the crime scene as well as sample number 2 or per suspect number 2. And based on this, you can kind of discern that suspect number 2 is involved in the crime scene. That's a really simplified way of how you can kind of compare our DNA samples and how it can be used in the real world. But let's just review in general what we learned today. So the function of DNA gel electrophoresis is to separate DNA based on size, and it helps us analyze genes and compare DNA samples. DNA is negative because it has a negatively charged phosphate ion in its backbone, and therefore when you put it in our gel electrophoresis apparatus that has a electrical gradient from negative to positive, our DNA will move across the gradient. And the reason why we can separate our DNA based on size is because larger strands take a longer time to move across the gel and through all of the tiny pores in the gel. And therefore you can have a separation between larger strands and smaller strands of DNA. In the next video, we're going to be showing you how to do this procedure, how to create your gel, how to create your samples, and provide a physical example of how your gel should look. Well, thank you for listening, and I'll see you guys on the next video.